Hello, good afternoon, dear students. I'm sure that I'm audible to all of you. Now I will switch off my this video and would like to share with you my screen. So today topic is oxidative phosphorylation and electron transport chain. We shall be discussing all these in the two parts. First part, I will discuss about the electron transport chain today. But before discussing to electron transport chain, I would like to recall our last lecture, what we have learned so far. We have learned so far that what is entropy, what is enthalpy, what is free energy, and how they are interacting in a system and surrounding, and how come the different bonds are important, what is the water doing in it, and ultimately, whatever the initial state and the final states, energy levels are there, not the kinetics part, which you have already discussed in the enzyme section, that is called bioenergetic. But purpose of all is to sustain the all the cells of our 72 trillion cells, whether the bacteria or viruses or bee, all are having the machinery to generate the energy, right? And this energy need to be generated in a such a way that the all activities are sustained and life goes smoothly. But the problem is when this energy is overproduced or during this energy production some machinery is defective, then there are many diseases, right? So in all basically aerobic organisms, they are able to capture far greater proportion of the available free energy of respiratory substrate than the anaerobic microorganisms. Most of this takes place inside the one organelle. All of you are aware about that is called mitochondria. And this mitochondria, because this is participating in the generation of energy, this is called powerhouses. Basically, the purpose of this one use the oxygen, produce the ATP. That's why the respiration word is used. During the respiration, it is coupled with the generation of the high energy intermediates, like the most important, the ATP. Hence, the respiratory chain which is happening is coupled with that one, that is called oxidative phosphorylation. Why this is important to understand? Because you are going to become a doctor, and many of the drugs used for the treatment and certain poisons may be affecting this electron transport chain or respiratory chain or oxidative phosphorylation. For example, I'll be coming to that one, but in the beginning, I'm telling you barbiturates, cyanide, carbon monoxide. These are essential to know that where they are affecting and how they are affecting. That will affect the mode of the treatment and the survival of the affected person. Not not only this, there are many inherited defects in the mitochondria, which we shall be discussing in the subsequent lecture. For example, myopathy, encephalopathy, and lactic acidosis. To understand this first part of oxidative phosphorylation, let us start with the electron transport chain. We are eating lot many compounds, but the energy-rich molecules such as glucose, they are metabolized by a series of oxidation reactions, and they are ultimately yielding carbon dioxide and water. Our food is in the form of carbohydrate, fatty acids, amino acids. But our concern is they are basically converting into carbon dioxide and water. But what they are producing? NAD, nucleotide, and FAD, which are converted into NADH and FADH2. And proton is generated, hydrogen ion is generated. So these NADH and FADH2 are further, these basically coenzymes they are transported, and because of due to some mechanism which I will be discussing with you, they are going to produce a ATP. So the metabolic intermediates of these reactions donate electrons to specific coenzymes like NAD and FAD. They form the NADH and FADH. These reduced coenzymes, NADH and FADH2, in turn each donate a pair of electrons to a specialized set of electron carriers, and these specialized set of carriers are collectively called electron transport chain, right? And when they are coupled with oxidation, they are called the oxidative phosphorylation. So I'm sure that I have told you during the metabolism, the purpose is whatever you are eating, nutrients, they are ultimately metabolized and producing certain oxidants which are reduced and producing the reduced coenzymes, and they are passing through a set of carriers. They are called the electron transport chain. Oxygen is required for that one to convert them into carbon dioxide and water. Hence, they are called oxidative phosphorylation. These electrons are passed down to the electron transport chain. They lose much of their free energy, and part of this energy can be captured and stored by the production of ATP. How they are produced? ATP is produced from the ADP combining with the inorganic phosphate. Apparently, it looks very easy, but it is a tough process, right? I'll explain each and every step of that one. Some energy is converted by this one into the ATP, but the remainder of the free energy, which is not trapped as ATP, is used to drive other activity. They are called the ancillary reactions, such as calcium transport into mitochondria and to generate heat. This generated heat will maintain our body temperature. As I told you, mitochondria is basically powerhouse. So let us understand about the mitochondria. Structure you have already read in your 12th standards, but I will explain you. The electron transport chain is present in the inner mitochondrial membrane. If you see in the diagram clearly, there is an outer membrane, there is an intermediate space, and then the cristiforms, convoluted tubules like things are there, means cristiforms are there. That is the inner mitochondrial membrane. And on this, there is a final common pathway by which electrons derived from different fuels of the body flow to the oxidation. By means oxygen, where this is called oxidative phosphorylation, means whatever is there. So outer membrane, intermediate space, then inner membrane, and in between there is called matrix. So electron transport and ATP synthesis by oxidative phosphorylation proceeds continuously in all tissue that contains mitochondria. The major components of the electron transport chain are located in the inner membrane. Basically, if you when you will study, there are different types of enzymes. For example, in the glycolytic pathway, when you will see in the cytosol, there are many enzymes here and there, and they are participating in the reaction. They are not organized at all, right? And certain enzymes, if you see that like the EST is present in the cytosol as well as in the mitochondria, means those who are present and not organized are called poorly organized, and if they are present in the two compartments, they are called compartmentalized enzymes. If when you will study about the triacylglycerol synthesis, then you will come to know that one enzyme complex is there, where the 7-8 enzymes are there. They are arranged into head to tail arrangements. So they are called highly organized. But beauty of this enzyme system present in the electron transport chain is they are present in the inner membrane and in particular fashion. So you can say this is the highest organization level of the enzymatic system. Why? Because this way, once the process has started, it will produce to the ATP until unless In between, something goes wrong by something, by some drugs or the poisons, they will be called inhibitors, right? Or other mechanisms by which these reactions are not coupled. If they are not coupled properly, then they are called the uncouplers, which I'll be discussing later. The outer membrane contains special pores, they are called the porins, making it freely permeable to most ions and small molecules. But the inner mitochondrial membrane is a specialized structure that is impermeable even to hydrogen, sodium, potassium in small molecules such as ATP, ATP, pyruvate, and other metabolites important to mitochondrial function. The purpose is there behind it. In the body, anything has been designed in such a way that until unless you understand the intricacy of it, you will not be able to understand why it has been designed. But this is beautifully designed inner mitochondrial membrane. What happens? Specialized carriers or transport systems are required to move these hydrogen, sodium, ATP, ATP, and all these are, and they are present in the inner membrane. Move ions or molecules across this membrane. The inner mitochondrial membrane is usually rich in protein, half of which is directly involved in the electron transport and oxidative phosphorylation. How many proteins? If you recall from the genetics mitochondrial DNA properties, you will come to know that is a roughly 695 base pairs circular DNA. And the beauty of this DNA is that it comes from the only from the maternal side. And this produces, most important, almost 69 proteins are required for the normal functioning of the mitochondria for this electron transport chain and oxidative phosphorylation. But the lucky 13 proteins are synthesized by only mitochondrial DNA. Rest proteins are imported from the cytosol, from the nuclear DNA. This information I'm adding because then you will come to know that why I'm saying that unusually rich in protein, but half of which are directly involved in the electron transport and oxidative phosphorylation. The inner mitochondrial membrane is highly convoluted. They are called cristae. This serves to greatly increase the surface area of the membrane. You know that anything, if you keep on folding, that will increase the surface area. Same way, the cristae are increasing the surface area of the membrane. And the most of reactions which are happening, like TCA cycle, fatty acid oxidation enzymes, mitochondrial DNA or mitochondrial RNA, mitochondrial ribosomes, they are present in the matrix. What is matrix basically of mitochondria? This is gel-like solution in the interior of mitochondria, which is almost 50% protein. And these molecules include the enzymes responsible for oxidation of pyruvate, amino acids, fatty acids by beta oxidation, TCA cycle. The synthesis of glucose, urea, and heme occur partly. When you read the urea cycle, partly it happens in the matrix and partly in the cytosol. Heme synthesis, they are present occurring in the mitochondrial matrix. The matrix contains basically our useful thing is NAD and FAD, the oxidized form of the two coenzymes that are required as a hydrogen accept, right? ATP and inorganic phosphate, which are used to produce the ATP. The matrix also contains mitochondrial RNA, DNA, and ribosomes. So our concern today is that mitochondrial matrix contains NAD, FAD, ADP, and PI. So what happens? The inner mitochondrial membrane can be disrupted into five. Means I'm disrupted word using is if you see this organized enzyme system, you can per se they are not divided in complex 1, 2, 3, 4, or 5. Don't think like this one. But for understanding, we can disrupt them into 5 parts, right? This is for only understanding purpose, but otherwise this diagrammatic presentation is not like that one, that you may have the impression that yes, it looks like this and it works like that. But no, for understanding, we can say there are the 5 complexes. So complex 1 to complex 4, each contains part of electron transport chain, and each complex accepts or donates electron to relatively mobile electron. Just I'll give you the simile. What happens if you just go the electricity turbine somewhere? What happens? Water is falling from some place to then another step, then another step, then a step, and then there is a turbine which keeps on revoluting. So you can say that 4 5 steps from where water is falling. There are complex 1, complex 2, complex 3, and complex 4, and where the revolutionary turbine is basically a complex 5, which is ultimately producing the ATP. Whether really it is producing ATP or doing something else, I will share you later. So, organization of the electron transport chain, if you see, each carrier in the electron transport chain can receive electrons from an electron donor and can subsequently donate electrons to the next carrier in the chain. Means you have to pass the pattern, one electron to transfer to another carrier, then another carrier, then another carrier, and fire it to the final turbine. These electrons ultimately combine with the oxygen and protons to form water molecule, right? This requirement of oxygen makes the electron transport process the respiratory chain. That's why what is it called? Electron transport chain or respiratory chain, then finally oxidative phosphorylation. Don't get confused. Which accounts for the greatest portion of the body's use of oxygen. Normally we think that we are breathing the oxygen and oxygen is just going with the hemoglobin and to the different cells. What is the basic purpose of this oxygen is that every cell is using oxygen to generate most of that one is to produce the ATP, right? Complex 5 is a protein complex that contains a domain. I will discuss with that one, but I am giving the overview before that one, giving the complex 1, 2, 3, and 4. There are parts, they are just like a turbine. I will try to show you the video of this one, how it works in the next lecture. But they are basically in the two domains, F0 that spans the inner mitochondrial membrane, and domain F1 that appears as a sphere that protrudes in the mitochondrial matrix. So complex 5 catalyzes ATP synthesis and so is referred as a ATP synthase. Now let's start the reactions of electron transport chain. I'll show you first the total diagram. Let me. If you see, there are 4 steps are there: complex 1, complex 2, complex 3, and complex 4, right? So what we have to do, we have to start means transfer the electrons up to formation of the water molecule. So with the exception coenzyme Q, if you see in the all, in one line I'm trying to say, with the exception of coenzyme Q, all members of the chain are proteins. And these proteins are very important to understand. They may function as an enzyme, as in the case of dehydrogenases. They may contain iron as a part of iron sulfur center. They may be coordinated with a porphyrin ring as in the cytochromes, or may contain copper as does in the cytochrome A and A3 complex. I will come each of them. The most important is, in the beginning I said, from the fuel molecules, whether it is carbohydrate, amino acids, or with the beta oxidation, you are getting ultimately NAD and FAD. So first let us see how the NADH is formed. NAD plus is reduced to NADH by dehydrogenase that removes 2 hydrogen atoms from their substrate. So both electrons, but only one proton. What is beauty in this reaction is 2 electrons, both electrons, but only one proton, that is hydride ion, are transferred to NAD and forming NADH plus a free proton, right? Because one hydrogen is going and attaching NADH, but one proton will be left. This NADH dehydrogenase, the free proton plus the hydride ion carried by NADH are next transferred to NADH dehydrogenase, a protein complex. It is not simple, it's this little tricky thing is a complex embedded in the inner mitochondrial membrane, that is a complex 1. This complex 1 has a tightly bound molecule like this one, FAD, flavin mononucleotide, FN. A coenzyme is structurally related to FAD that accepts the two hydrogen atoms, 2 electrons plus 2 hydrogen ion, becoming FMNNH2, right? This reaction is there, just depiction is there, you can read in any textbook. Riboflavin is there, flavin MM, ATP, ADP, then it is converted into PI, ATP, and FAD is produced, right? NADH dehydrogenase also contains iron atom paired with the sulfur atoms to make iron sulfur center. Means NAD is not directly converted to NADH, rather it goes from step to step. NADH, what happens is iron atom paired with the sulfur atoms to make iron sulfur centers. And these are necessary for the transfer of the hydrogen atom to the next member of the chain. Ultimately, you are going to pass the baton to the other one. Here, the next baton is to coenzyme Q, that is called ubiquinone. This is the only which is a non-protein. Coenzyme Q is a quinone derivative with a long hydrophobic isoprenoid tail. It is also called ubiquinone because it is ubiquitous in biological system. This is present in each and every cell. Coenzyme Q is basically mobile carrier and can accept hydrogen atoms both from NADH and FNH2 produced on the NADH dehydrogenase complex 1 and from FADH2 produced on the succinate dehydrogenase complex 2, glycerophosphate dehydrogenase and acyl-CoA dehydrogenase. Means it can accept hydrogen atoms from anywhere. If you see the diagram, NADH means the dihydroxyacetone phosphate to glycerol 3 phosphate, and this glycerol 3 phosphate to again dihydroxyacetone phosphate. In the inner mitochondrial membrane, FAD is converted to FADH2, and where it is going? Coenzyme Q of the electron transport chain, right? And if you see from the these other sources, they are also in the same form. Coenzyme Q transfers electron to complex 3, and this coenzyme Q then links the flavoproteins to the cytochromes. Now I am coming to the cytochromes. The remaining member of the electron transport chain are cytochromes, and each contains a heme group, a porphyrin ring plus iron. Unlike the heme, but heme groups in the hemoglobin, the cytochromes, what is special is the cytochrome iron is reversibly converted from its ferric to its ferrous form as a normal part of its function as a reversible carrier of electrons. These electrons are passed along the chain from coenzyme Q to cytochrome BC1 complex 3, C and A plus A3 complex 4. Note here that cytochrome C is associated with the outer face of the inner membrane, and like coenzyme Q is a mobile carrier of electrons. Cytochrome A and A3. This cytochrome complex is the only electron carrier in which the heme iron has an available coordination site that can react directly with oxygen. Because this can directly react with the oxygen, it is called cytochrome oxidase. At this site, the transported electron, oxygen, and free protons are brought together, and oxygen is reduced to water. Here, if you can see, of half oxygen, water, this iron pays to ferric, the complex 4, by this is cytochrome C oxidase, right? Cytochrome oxidase contains copper atoms that are required for this complex reaction to occur. Now, as I told you in the beginning, during the significance itself, so far we have understood that these electrons has to pass through the four complexes, and finally fifth complex they will be going, right? In between, there are some drugs are there which can inhibit the steps. Just see here, means NAD to FMN, OUQ, cytochrome BC1, cytochrome C, and cytochrome E3, right? So inhibitors prevent the passage of electron by binding to a component of the chain, blocking the oxidation reaction. Therefore, all electron carriers before the block are fully reduced, and whereas those located after the block, they are the oxidized. So inhibition of electron transport inhibits ATP synthesis because these processes are tightly coupled. If you see at this stage, which one is amytol and rotenol? If at this stage, if you see the antimycin A, this is here. At this stage, cyanide, carbon monoxide, and sodium azide. Why this is important? And I'm keeping it for a longer time. Just have a look, though it is copied from textbook only. Nowadays, the question, you are having the multiple choice questions, right? And when you will be appearing in the examination, similar kind of diagram will be there, right? Suppose I have to ask rotenone blocks at which stage. Then I will write 1, 2, and 3, and one more site I can make anywhere, right? Then you have to choose at which stage. So diagrams are very, very important nowadays to be picturized, visualized properly. So there will be rotenone will be removed, and which blocks, which inhibitor blocks at this stage. Similarly, which antibiotic inhibits at this stage. You have to write the antimycin. Then I can ask the question, cyanide blocks at the site 1, 2, 3, right? So this kind of diagrams you have to remember. Blocking electrons transfer by any one of these inhibitors, as I told, what inhibit? ATP synthesis. Now, the most important thing is, if inhibitors are there are by any mechanism, oxygen is not converted into water, because ultimately you are doing what will happen means incomplete reduction of oxygen to water produces reactive oxygen species such as superoxide, hydrogen peroxide, and hydroxide radical. Basically, in our body, oxygen is a two-base word. If oxygen is not sufficient, hypoxia is there, then there is a problem. If anoxia is there, there is a problem. And if oxygen is not properly reduced, even then there is a problem. In the body, there are enzymatic systems to combat this, and there are the non-enzymatic systems are there. But for the reactive per se, for the reactive oxygen species, enzymatic system like superoxide dismutase, catalase, glutathione peroxidase enzymes are there, which will take care of these superoxide, hydrogen peroxide, hydroxide radicals respectively. So these reactive oxygen species, what they do basically, they affect the DNA and proteins, and apart from this, they produce the lipid peroxidation. Total mechanism will be taught somewhere when the case studies will be taken care. So enzymes superoxide dismutase, catalate, and glutathione peroxidase are the cellular defense against the reactive oxygen species. This is such important that there can be a complete lecture on the reactive oxygen species and free radicals, because from cancer, aging to apoptosis, or weight loss, obesity, diabetes, gestational diabetes mellitus, preeclampsia, many diseases are associated with generation of reactive oxygen species, right? Let's come to see how the energy is released from the electron transport chain, right? The free energy is released as electron are transferred along the electron transport chain. We have understood so far from an electron donor, reducing agent or reductant, to an electron acceptor. Means total story is simple. Someone is donating and someone is accepting, and finally oxygen is converted to water, because oxygen is the final acceptor of these electrons, right? These electrons can be transferred as, for example, in NAD, in what form we are transferring? As a hydride ion. In FMN, as a hydrogen atom. In coenzyme Q and FAD, an electron to cytochromes, right? Now, when these reactions are happening, they are happening in a pair, right? Means oxidation, there is a loss of electron of one compound is always accompanied by the reduction of a second substance, means gain of electrons. So that's why overall means oxidation reduction is going together. So redox pairs are formed, means NADH, hydrogen and then NAD plus. Similarly, FMN is converted to FNH2. And another one is, if you means if a pair are there, means you can really pair them, means NADH plus half of the reaction you can write half like this, means NAD plus 2 electrons plus 2 hydrogen, redox pair. Another redox pair will be FMN 2 electron 2H, but only this one we convert to FMN H2. So example, the oxidation of the NADH to NAD accompanied by the reduction of the FMN to FNH2. Such oxidation reduction reactions can be written as a sum of two separate half reactions, one an oxidation reaction and the other reduction reaction, right? We have already discussed. But these redox pairs differ in their tendency to lose electrons. And this tendency is a characteristic of a particular redox pair. For example, in this redox pair, and this can be quantitated also. So quantitatively specified by a constant is, this is called the standard reduction potential. The unit for that one will be the volt. So for this NADH to H, NAD redox pair is, here the standard reduction potential is minus 0.32 volt, and here it is a minus 22 volt, right? This standard reduction potential importance. Let's see. The standard reduction potential of various redox pairs can be ordered from the most negative to the most positive, right? And you know the importance of negative and positive as in delta G. Here also it is important. The more negative the standard reduction potential of a redox pair, the greater the tendency of reductant member of the pair to lose the electron. Other way around, the more positive the standard potential of redox pair, the greater the tendency of the oxidant member of the pair to accept the electron. Therefore, electrons flow from the pair with the more negative standard reduction potential to that with the more positive standard reduction potential. The components of electron transport chain are arranged in order of increasingly positive standard reduction potential. Here NADH, NAD and NADH is minus 0.32, FMN, FNH2 is minus 22 volt, cytochrome, ferric to ferrous 0.22 plus, and oxygen finally is having the 0.82. Compound at the bottom of the table are a strong oxidizing agent, oxidant, that is they accept electron, right? Now let's relate this standard E0 with the delta D. The change in free energy is related directly to the magnitude of the change in the E0. How they are equated? Delta G0 is minus NF. And what is F basically here? That is called Faraday constant. N is the number of electron transferred, 1 for a cytochrome, 2 for the NADH, FADH2 and coenzyme Q. Means cytochrome transfers how many electrons? 1. And NADH, FADH2 and coenzyme Q transfer the 2 electrons. E0 of the electron accepting pair minus E0 of the electron donating pair is called delta E0. And delta G0 we know the change in the standard free energy. Now this delta G0 of ATP, you know all of you that The standard free energy of phosphorylation of ADP to ATP is plus 7.3 kilocalorie per mole. Why I am going to talk about this one? Because this is a very common question in your Viva boards, that how much ATP is produced by this system, how much ATP is produced by this way, that way. So to understand that one, it will be easy if you just understand this one. Just a minute, give me a moment. Right. Means finally, when I'll teach you in the subsequent class, in the complex one, or right now you can understand, suppose NADH is going, what will happen? There will be number of electrons will be transferred, right? The delta G0 for the phosphorylation of ADP to ATP is 7.3 kilocalorie per mole, right? The transport of pair of electrons from NADH to oxygen through the ATC releases how much ATP? How much energy? 52.6 kilocalorie. How? By that formula delta G0 minus NF delta E. Basically, N will be the 2 in number, then multiply with the Faraday constant 23.1, then multiply it with the delta E0 by 1.4. Then the total number of protons formed out of inner mitochondrial membrane is almost, if you see what will happen if they are going through the complex 1, complex 3 and complex 4, 4 protons at first complex, 4 protons at the third complex and 2 protons at the fourth complex, means 10 protons are produced. For synthesis of 1 ATP, average number of protons that need to enter through complex 5, which I shall be discussing tomorrow. Sorry, before that I am telling you the calculation, so there may be little confusion. Just accept it that there will be the only 4 protons will be required, right? For transport of pi to mitochondrial matrix, 1 hydroxyl ion goes out of matrix by pi transport, and this equivalent to entry of the 1 proton. So this way, synthesis of 1 ATP, 4 protons enter into the matrix. Thus, 1 NADH produces 2.5 ATP, while 10 protons versus 4 protons. Proton 10 are going out and 4 in, means 10 divided by 4. How much ATP? 2.5 ATP. Sometimes expressed, these equations are sometimes expressed as the PO ratio. So ATP made per oxygen atom reduced of 2.5. Energy captured as a currency of energy. So how much it will become? 18.25 kilocalorie. And this efficiency of an energy capturing system is calculated by using equation. You can write it on pen and paper. Efficiency is equal to energy captured divided by energy released multiplied by 100. So if you see the efficiency of oxidative phosphorylation when 1 NADH forms 2 ATP, it comes roughly 34.7%. The remaining calories are used for the ancillary reaction. In the beginning, I told you, some of them will be converted to the ATP, rest will be participating in the ancillary reactions or release of the heat. And suppose you go by the FADS2, then it is not going to pass through the one complex. So what will happen? Only 1.5 ATP it will be produced. So 1.5, 1, because complex 1 is bypass in this, right? So calculation, again, if you do, the total proton pumped out will be 4 plus 2, complex 2, 4, and complex 3, 2. So it will come the 6, and total proton pumped in 1 ATP synthesis will remain same. 3 plus 1 means 4. So divide 6 by 4. How many ATP will be generated? By the FADS2, 6 by 4, 1.5, right? Now, This mitochondria, suppose mitochondria process in the mitochondria, carbon monoxide, cyanide, and hydrogen sulfide are inhibitors of electron transport. As I told you, cytochrome C to oxygen. Carbon monoxide inhibits the enzyme cytochrome C oxidase of the human mitochondrial respiratory chains. This carbon monoxide poisoning is a serious risk whenever the carbon monoxide is released in a closed space like a closed car. That's why whenever you are going and starting your car or using for longer times, it's better to open your windows for some time because there will be close so much production of the carbon monoxide. Or sometimes we lock our air conditioning room for very long duration. Again, there is a carbon monoxide generation is there. So avoid it, open room for some time. So sleeping in the packed car with the engine on, AC running, is cause of death due to carbon monoxide poisoning. What are the symptoms for carbon monoxide poisoning? There is dizziness, weakness, confusion, and can progress to arrhythmias, seizures, and ultimately to death. OXPHOS diseases are there, and the clinical disease involving component of oxidative phosphorylation. They may be caused by mutations in a nuclear or mitochondrial DNA. As I told you, certain proteins are produced in the mitochondrial DNA, and some of them are imported from the nuclear DNA. Other inhibitors of electron transport chain target complex 1, 2, 3, 4, ATP synthase, and ATP-ADP antiporters also. Rotenone, I have already told, rotenone is very common. Basically, where it is used it is used as a pesticide in the farming. It inhibits basically aerobic cellular respiration by blocking NADH dehydrogenase. And exposure to potassium cyanide can be rapidly fatal. Potassium cyanide, I have told you that at which stage this is participating. This is important because poisoning of potassium cyanide is basically not well established because it is too fatal that what features are going to develop. It is difficult to understand. But this is a deadly poison. After understanding bioenergetics, the keywords which we have learned yesterday: free energy, enthalpy, entropy, system surrounding, and now in the biological system, how these oxidants are converted into reductant, and finally oxygen is producing the water. They will be coupled with the oxidative phosphorylation. That part we shall be discussing tomorrow, right? I could not see your questions yesterday, that who has asked the questions. If you are having any query, you can put it later on after this one. Today I am giving you this much information because too much information is also not good. But if any query in your mind comes, please feel free to contact me personally or on the WhatsApp, please. Thank you very much.